Hey guys, it's Mrs. Esser. Today we are going to be starting our protein synthesis unit, but before we get into the two steps of protein synthesis, transcription and translation, we really need to make sure you guys have a full understanding of DNA versus RNA. So in this video, we're going to take a look back at nucleic acids, which should hopefully be reviewed because this was one of our 4 macromolecules within our biochemistry unit. So, some reminders with our nucleic acids, our building block is called a nucleotide, which is made up of a phosphate attached to a pentose sugar, pentose because there's 1, 2, 3, 4, 5 sides, and then a nitrogen base. So, the two different types of bases we could have are purines or pyridines, and we'll talk about those a little bit more later. But our nucleotides or our nucleic acids are used for genetic information, or we could say hereditary information, how traits are passed down from one generation to the next. And we're going to be looking at how DNA replicates itself later on this year. But today we're going to be looking at how we take our DNA, how we create mRNA, and how that's going to transform into a physical trait for your body. So some things to remember with our nucleic acids. Building block is a nucleotide. Remember, monomer means one. Our RNA is single-stranded and our DNA is double-stranded. So I want to dive in and take a peek at RNA and DNA right now. So with our DNA, remember our DNA is very, very long strands of genetic information. So every single cell in your body has its own set of DNA that is completely identical. Most humans' DNA is very much alike. Your DNA and my DNA is probably 99.7% the same. The difference is there's these small segments of DNA called a gene. So, a gene, much like a light switch, can turn on and off. So, perhaps you have a gene that codes for brown eyes that's turned on. And for me, that gene is turned off because my eyes are green. So, a gene is a small segment of DNA that can be turned on and off that will end up coding for your physical traits. So we've already seen this slide before, but with our deoxyribonucleic acid, remember we think of it as the queen bee. The queen bee never leaves her hive. Same thing, our DNA will never leave the nucleus. It's stuck inside of it and it's too big to get out. We say the DNA is too big because it's the shape of a double helix or a twisted ladder. So I can see the backbones are made up of sugar and phosphates. So remember, you want a strong backbone. So our DNA's backbone will be strong as well. On the inside, we have our nitrogen bases, which in DNA are thymine, cytosine, guanine, and adenine. So our DNA, double helix, shape of a twisted ladder. Reminder that the sugar and phosphates are connected by something called a phosphodiester bond, which is an extremely strong bond. So the backbone should never come apart. The nitrogen bases in the middle, we have A pairing with T. Remember, apples in the tree. C always pairs with G, cars in the garage. Between our nitrogen bases, we have these hydrogen bonds, which remember, hydrogen bonds are actually really, really weak bonds. So thinking of them like a magnet, I can put a magnet on my fridge and I can take it back off. So a hydrogen bond, I can put my nitrogen bases together, but I can pull them apart because we're going to have to separate our DNA in order to, one, replicate it, in order to, two, express our genes or to code for those proteins. So all of our nitrogen bases are purines. Remember, purines, pure as gold. Purines are adenine and guanine. And the pyrimidines are cytosine and thymine, which are present in DNA. And then uracil and cytosine are present in RNA. So we have this guy named Chargaff who came up with the base pairing rule in DNA. He said A always pairs with T, C always pairs with a G. So apples in tree, cars in the garage. One thing I didn't teach you guys yet is talking about DNA being anti-parallel. So if I am driving one way down the road and another car is driving the opposite way as me, we're going to cross paths. So that's considered anti-parallel. We're going opposite directions from one another. So our DNA is considered anti-parallel, and how we tell that is we always look at the sugar. So I'm going to start focusing on the sugar on the left side here. So this sugar phosphate backbone, notice the oxygens are all pointing up. So sometimes I think of the oxygen as zero because we count the carbons to know what direction my sugar is going. So we count clockwise. So I have zero, carbon one, two, three, four, and five. Every sugar on this backbone will be the same: zero, one, two, three, four, and five. So I say this sugar phosphate backbone is running in the 5' to 3' direction. You'll notice on the other side, my oxygens are pointing down. So we still always count clockwise. So I have zero, one, two, three, four, five. And it'll be the same on every single sugar on this side. So we say this backbone is running in the 3' to 5' direction because reading top to bottom, I see 3, then 5 first for every single one of my sugars. I show my bases here, my nitrogen bases. G goes with C, these weak hydrogen bonds. That's why it's not a solid line, they're just dashed. And apples in the tree, adenine and thymine, connected by my weak hydrogen bonds. Some history of DNA, just some scientists to know. Erwin Chargaff, he's the one who came with Chargaff's rule: A pairs with T, C pairs with G. Rosalind Franklin, she was actually the first person to take a picture of DNA. And unfortunately, she died shortly after, so she was not able to publish her findings. But James Watson and Francis Crick took her findings and published them. So they're actually the ones who are credited for finding the structure of DNA and saying that DNA is a double helix. So switching over from DNA to our RNA. RNA, I want you to think of as the drone bee or the worker bee. So if DNA is the queen bee, never leaves the nucleus. Our drone bees are the ones who are doing all the work. They're going around, they're sending messages, they're collecting things. So we're going to see in this unit how ribonucleic acid or RNA is like a drone bee. So still sugar phosphate backbone, but instead we're going to have our sugar ribose, which is where ribonucleic acid gets its name. And you're going to see instead it's a single helix, which makes ribonucleic acid smaller. And because of that, RNA is able to go in and out of the nucleus where DNA would get stuck. The nitrogen bases are mostly the same. We have adenine, guanine, cytosine, but notice we have uracil in the place of thymine. So wherever you'd normally put a T in DNA, you're going to put a U for uracil in RNA. So here I can see my base pairing rule. A goes with U now, and G goes with C. So what we're going to see in our protein synthesis is the DNA opens up, and based off one side of our DNA, the mRNA is going to be created. So if I see an A on that DNA, a U is going to come and take its place. So we have three different forms of RNA I want to look at: messenger RNA, ribosomal RNA, and transfer RNA. So messenger RNA, mostly written as mRNA, is literally a message. So we take our DNA, we separate it, we create the mRNA. So it's going to make a message from a small segment of DNA, also known as a gene. So if the gene is turned on, the mRNA, the message will be taken from that DNA. And that mRNA is going to leave the nucleus through those small nuclear pores, and it's going to bring this message to the ribosome. So the ribosome is actually the site of protein synthesis. It's where proteins are made. So the ribosome is going to use the message from the mRNA to create a protein in order to express that gene or express that trait. Our second type of RNA is ribosomal RNA, mostly written rRNA. And what I want you to know for Bio 1 is it just builds a ribosome. It's where the mRNA comes to attach to. The tRNA comes there as well to drop off the amino acids. So ribosomal RNA makes the ribosome. Our last one, our transfer RNA or our tRNA, carries the amino acid. So you can see the amino acid at the top here. So the tRNA will pick up the amino acids in the cytoplasm and it will bring them to the ribosome. So we'll talk about it in translation, but there's this message down here so the tRNA knows when to land on the ribosome. When it does, it will drop off the amino acid and then it will take off to go pick up more amino acids. So also review from biochem, we should know that amino acids link together to form a polypeptide chain. When I fold my polypeptide chain, I'm going to make a protein. Remember, protein's structure equals function. So the structure and shape of that polypeptide chain will determine the function of what that protein does for the body. So for my visual people, here are just some differences between our DNA and RNA. RNA, I can see my single strand. DNA, I have two sugar phosphate backbones. DNA, there are thymines, whereas RNA, there are uracils in its place. And then I have ribose in place of deoxyribose for my RNA. Then our last slide, just looking at function differences between DNA and RNA. DNA, it's going to carry the gene, it's going to carry the code for the polypeptide, so it carries all of our genetic information. Whereas the RNA, it's going to rewrite, it's going to translate that message from the DNA so the ribosome knows how to use it, and the RNA is going to in turn make that protein. So that's it for DNA versus RNA. On our next videos, we'll be looking at protein synthesis, which is called transcription and translation.